Hello, this is Professor White, and I'll be teaching mitosis and cytokinesis, particularly your cell cycle. Now, our key concept today is cells have distinct phases of growth, reproduction, and normal functions. So your goal throughout this lesson is for you to summarize the characteristics of cell cycle interphase called G1S, and G2 phase, the phases of mitosis called prophase, metaphase, anaphase, and telophase in plant and animal cytokinesis. Now, the cell cycle is a repeated pattern of growth and division that occurs in the eukaryotic cells. Now, the cell consists of three phases, G1, S, and G2. Now, the first phase represents the cell growth, while the last phase represents the cell division. Now, the cell cycle has four main stages. Now, during the cell cycle, the pattern is for the cell to grow, then the DNA replicates, and of course, the cell will divide. Now, as you can see here in this diagram, most of the cell's life is devoted to interphase. As you can see, most of its cycle is in the interphase. Like the G1, this is where the cell grows, carry out different normal functionings of the cell, and replicate their organelles. Now, the synthesis phase, or S phase, is when the DNA starts replicating. As you know, replication of DNA signals that the cell will have to make its own kind with the same DNA. And of course, the G2 phase, or the growth 2 phase, this is pretty much an additional growth that the cell needs. And of course, right after the interphase comes the mitosis phase, which is your cell division phase. And we're going to discuss that today. Now, the main stages of the cell cycle are gap 1, synthesis, and gap 2, and mitosis, which we already know. And like I mentioned, this is where your cell is growing and functioning as it is. The S phase is when the DNA copies itself, and the G2 is the additional growth, wherein chromatids become replicated chromosomes. And the mitosis includes the division of the cell nucleus and the division of the cell cytoplasm. Now, mitosis only occurs if the cell is large enough and the DNA is undamaged. Now, let's take a look at the interphase. Now, the cell, like I mentioned earlier, spend the majority of their cell cycle in the interphase. Now, the purpose of the interphase is for cell growth. Now, by the end of interphase, the cell has 2 full sets of DNA or chromosomes and is large enough to begin the division process. Now, this is an image of your chromosome, and each of the sides are sister chromatids. And basically, your chromosome is composed of supercoiled DNA and has beads around it called histones. So this is a great image for you to look at, and your DNA is really tightly coiled in your chromosome. Now, chromosomes condense at the start of mitosis. And as I mentioned earlier, your DNA is super coiled in your chromosome. And if you unwound them, you will be able to see the histone protein. And if you unwound a little bit more, you will see your DNA as a double helix right here. So the DNA wraps around proteins, which are called histones, that condense it. And in a typical human cell, there is about 6.5 feet of DNA. Now, here's a diagram that shows you what chromatids are. Chromatids is like one half of your X. The one that's in the middle of the sister chromatids is called your centromere. And the sister chromatids are normally the X shape you see here. And homologous chromosomes are 2 sets of chromosomes. Now, what is mitosis? So the purpose of mitosis is cell division, making 2 cells out of 1. Now, each cell has to have its own cytoplasm and DNA, and the DNA is replicated in the interphase when 2 chromosome strands become 4 strands of DNA, so 2 strands per chromatid. Now in mitosis, the 4 strands or 2 sister chromatids have to break apart so that each new cell only has 1 double-stranded chromosome. So 2 sister chromatids together make a chromosome. Now, what's the reason behind cell division? Well, the cell size is actually limited. The volume increases faster than surface area. And in this diagram, I believe we've learned this previously, that the volume increases faster than the surface area, which is why the cell size is limited, which is why it triggers your cell division. Now let's go to prophase. Now prophase is characterized by 4 events. First, the chromosomes are condensed, they are more visible, and the nuclear membrane or the envelope is disappearing. Now the centrioles have separated and taken position in the opposite poles. I'll show you what it is. And the spindle fibers form and radiate towards the center of the cell. Now the DNA plus protein is called chromatin. Okay, and 1/2 of a duplicated chromosome is a chromatid. Now, sister chromatids are held together at the centromere. It's right here, okay? And telomeres protect the DNA and do not include genes. So that's your telomere, like those edge right there. Now, mitosis divides the cell's nucleus in 4 phases. During prophase, chromosomes condense and spindle fiber forms, as you can see over here. Okay, spindle fibers is right there. Your chromosomes are really condensed. Now, metaphase is the shortest phase of mitosis, and it's characterized by 2 events. The chromosomes line up across the middle of the cell, and spindle fibers connect the centromere of each sister chromatids to the pole of the cell. Mitosis divides the cell's nucleus in 4 phases. During metaphase, chromosomes line up in the middle of the cell, right here. Anaphase. The next phase is anaphase, which is characterized by 3 events. The centromeres that join the sister chromatids now splitting. Sister chromatids separate, becoming individual chromosomes. And separated chromatids move to the opposite poles of the cell. Now, during anaphase, sister chromatids separate to opposite sides of the poles. You can see over here, they're now separating. Telophase. Now, telophase is the last phase of mitosis, which consists of 4 events. Chromosomes, each consisting of a single chromatid, uncoil. A nuclear envelope forms around the chromosomes at each pole of the cell. And spindle fibers break down and dissolve, and cytokinesis begin. During this phase, the nuclei form and chromosomes begin to uncoil, as you can see here in this diagram. Now, cytokinesis is the division of the cytoplasm into 2 individual cells. The process of cytokinesis differs somewhat in plant and animal cells. In animal cells, the membranes form a cleavage furrow that eventually pinches the cell into 2 nearly equal parts, each part containing its own nucleus and cytoplasmic organelles. So you can see over here. So that's a cleavage furrow. So it's like rounded, and then the formation of the nucleus surrounding the chromosomes in both new cells. In plant cells, a cell plate actually forms. Now, mitosis and cytokinesis produce 2 genetically identical daughter cells. Now, interphase prepares cells to divide, and during interphase, the DNA is duplicated. Now, here's an example of the plant cell during telophase. So instead of cleavage furrow, it actually forms what we call a cell plate right here. Now, cells divide at different rates. Now, the rate of cell division varies with the need for those types of cells. As you can see here, in skin cells, its approximate lifespan is 2 weeks. So that's basically the rate at which the cell needs to divide. Red blood cells, 4 months. Liver cell, 300 to 500 days. Intestines or intestinal lining is about 4 to 5 days. And your intestine, muscles, and other tissues, 16 years. So this is basically the lifespan of the cell in each of these cells. Oh, and some cells are unlikely to divide, which will stay in G0. They just die. Diploid. So what is a diploid? Now, diploid in autosomal cells or body cells or any non-sex cells, a cell that contains 2 sets of chromosomes chromosomes. Now the number is represented as 2n. N is the number of chromosome. Now humans have two sets of 23 chromosomes for a total of 46 chromosomes. 2n, or 2 times 23, is 46. Now, in any given somatic cell, there are 22 pairs of regular chromosome and 1 pair of sex chromosome, as you can see here in our karyotype. This is actually called karyotype. Now, karyotype is a number and appearance of chromosomes. Now, what do you notice about the size, the centromere, telomere, sex, and the number? As you can see over here, they have the same size, same shape for your homologous pairs. Now, why are there 2 of each chromosome? Well, homologous chromosomes are 2 chromosomes that are the same. This happens because diploid organisms have 2 of each chromosomes. Now, each pair is a homologous pair. One of the homologous chromosome was inherited from individual's father and the other from the mother. Now, for example, the 2 chromosome number 1 are homologous. However, a chromosome number 1 and a chromosome number 2 are not homologous because they are different chromosomes. I hope that this lesson today about cytokinesis, mitosis, and cell cycle clears up any confusion about our topic for this week. And if you have any questions, please let me know. And I hope you enjoy the rest of your day. Have a great day, and I'll see you in class. Bye.